This topic covers the structure and function of organelles that are involved with the synthesis of macromolecules and lipids within the cell. This will include the nucleus, ribosomes, and the endomembrane system. All of these structures are interconnected, so they will be mentioned more than once as we go along. As we have said often, eukaryotes are defined by having their DNA enclosed in a membrane. This organelle is called the nucleus. The nucleus protects the DNA in the cells by regulating what can come in contact with the DNA. The nucleus has a double membrane with pores that allow proteins and enzymes to be brought into the nucleus and for RNA and other molecules to leave the nucleus. Inside the nucleus is a structure called the nucleolus. It is the site of ribosomal RNA or rRNA synthesis. The subunits of ribosomes are assembled there before they are transported back out into the cytoplasm. Ribosomes are composed of RNA and proteins. This rRNA is synthesized in the nucleolus. They are combined with ribosomal proteins that are synthesized in the cytoplasm and brought into the nucleus, where they are assembled with the rRNA into ribosomes. Ribosomes come in two parts or subunits. One subunit is larger than the other. These subunits are transported out into the cytoplasm, where the ribosomes take up their jobs of making proteins. A copy of the genes in the nucleus, or mRNA, is brought out of the nucleus. The subunits of the ribosome clamp down onto the mRNA. The ribosome moves along the mRNA, reading the code and making a protein from the code. As you can see in this micrograph, some ribosomes are embedded in a membrane, the rough endoplasmic reticulum. Some are floating free, so to speak, in the cytosol. The endomembrane system of eukaryotic cells is composed of the nucleus, endoplasmic reticulum, vesicles, Golgi apparatus. Some of the vesicles are specialized, and they're called lysosomes, and the cytoplasmic membrane. The double membrane of the nucleus is connected to another structure outside of the nucleus, but inside the cell cytoplasm. See, here it is in this diagram. It is called the endoplasmic reticulum, or ER. This is a site for enzymes that synthesize various molecules within the cell. If it is studded with ribosomes, it is called the rough ER because it appears rough in electron micrographs. Some mRNAs from the nucleus come directly to make some of the proteins. If it isn't studded with ribosomes, it is studded with enzymes that are not easily visible under an electron micrograph. These enzymes synthesize lipids and carbohydrates for the cell. Little blibs of membrane or vesicles will pinch off from the ER, carrying proteins and lipids and carbohydrates to the Golgi apparatus. The Golgi has enzymes for modifying macromolecules it gets from the ER. It may combine a lipid and a carbohydrate to make a liposaccharide, or it might combine a lipid and a protein to make a lipoprotein. It may modify macromolecules in other ways too. You can think of the ER as the factory that makes the parts for the cell, say sheet metal and nuts and bolts. Using that analogy, the Golgi would be the factory that uses the parts to make the finished product, say a car, from the sheet metal and the nuts and the bolts. Vesicles then pinch off from the Golgi and take the finished molecules to where they need to go. The nucleus, ER, and Golgi all work together to manufacture what the cell needs. Lysosomes are a specialized vesicle that pinches off from the Golgi. Hydrolytic or digestive enzymes are made in the rough ER and modified in the Golgi. Then they are pinched off in the lysosome, which protects the rest of the cell from their action. Lysosomes can fuse with other vesicles called vacuoles. Vacuoles contain a lot of different substances, but let's say one contains food to be digested. Okay, where's my cursor? There it is. Enzymes in the lysosome are exposed to the food when the lysosome and the food vacuole fuse, and the hydrolytic enzymes do the job without destroying the ER or the other cytoplasmic structure or the other important structures in the cell. The cytoplasmic membrane, like all membranes, is composed mostly of phospholipids in a bilayer, like it shows in this diagram. Eukaryotic cytoplasmic membranes also have steroids, which are four-ringed lipids. Cholesterol is an example of a sterol found in animal cells. They provide stability to the cytoplasmic membrane, which is vital for those cells, such as animal cells, that don't have a cell wall. Cytoplasmic membranes are selectively permeable and have transmembrane proteins for moving the big stuff in and out of the cell. Vesicles can fuse with the cytoplasmic membrane. They bring freshly manufactured phospholipids from the smooth ER and the Golgi. They bring freshly manufactured transmembrane proteins from the rough ER and the Golgi. Vesicles can also fuse with the cytoplasmic membrane when molecules are being exported. They can dump cellular waste, they can export enzymes to the outside of the cell, and so on.